Hello, today is really the last day of the sandbox. I would we will look at the sandbox for once more time, and I will discard it later. Maybe I will add it in a new project. And I know I still haven't added your request. Sorry. um, Also, I still see some requests telling me to add some of the old samples to the new samples. I think it is still worse, so I will keep some part of the samples and discard the remaining. I think I have added a rotten strawberry a few days ago. I can I can still show you because I still have one moldy strawberry. Very disgusting. But I think I will keep it to the new sandbox. It is a good material for the startup for the new sandbox. Oh, I think it is a good question because it is difficult to tell whether there are germs inside the sandbox by just looking at the microscope. We have to do further tests. I think it is interesting to do further tests to explore whether there are germs inside the samples. But I think I need to bring some part of the samples to the laboratory to do the test. It depends on how you define germs because some bacteria they are definite pathogens like salmonella. When you eat some salmonella, you will definitely get sick. But if you eat E. coli, you won't. Most of the time, you won't get sick. But if you eat large amounts of E. coli, you will get sick. So it really depends on how you define E. coli. And some bacteria, they only cause disease under certain circumstances. So I think it is interesting to look at what bacteria are surviving inside the samples. We cannot we cannot tell what bacteria are inside the samples by just looking at the microscope because they all look the same. But if I do further tests, we can differentiate them and we can look at whether there are dangerous bacteria inside. I think it is interesting. Yeah, I did read your comment. But I need to open the samples and take some out to look under the microscope. But you are right, it is really smelly, and I think I need to close the jar immediately. And you can still see the moldy strawberry are still here. Maybe I take the liquids around this moldy strawberry. Maybe there is something nearby, but I'm not sure. Maybe there are nothing. And actually, there are two types of microbes. Some like clean water and some like dirty water. So you will definitely see different kinds of microbes in dirty water and clean water. And for water bear like clean water, so it definitely cannot survive inside the sandbox like this. Okay, let's take a look. I just finished focusing. Can you see anything? But actually, 2 days ago, there's still many paramecia, but now I can't see any paramecia. Yes, you can. I also see many dots. We can zoom in. I'm using 100 magnification now. Really? You mean you saw one paramecia? I'm using 100 magnification now, so maybe I will zoom in later. But I want to give a brief look first. I want to look whether there are some large microbes there. My microscope is, let me think, 3 years old. I think the cam is dirty. I need to clean it first. Sorry. What do you mean old sample? Old sample. I have backup tubes of many days ago, like 40 days, something. We can take a look actually before I discard them. Anyway, they look the same, and never mind. Actually, you can see many things swimming. They are all bacteria. Actually, they are so small. I'm now using 400 magnification. For other microbes like paramecium, you can observe them clearly under low magnification, like 4T magnification. So you can see many rod-shaped bacteria swimming. Of course, I cannot tell the definite identification, the definite identity of this bacteria, but there's a high chance that it is Bacillus. Bacillus is a very common and ubiquitous bacteria that's living all around the environment, and you can find them everywhere, like on the air, on the table, on the skin. They are very common. So they are just contamination bacteria. They are not likely pathogen. But some bacillus can produce toxin that can cause diarrhea. And for this microscope, because I also add some other stuff like the phase contrast objective, so it is around, let me see, 5,000 USD. Yes, actually not very expensive. Okay, I see you, beef. I think I'm pretty sure that most microbes have died in the sandbox, so I'm not sure whether it is a good ending. But I don't want to make it so boring, so maybe I will take some old sample and we will look together later. Yes, I study microbiology, but I study medical microbiology, but not environmental microbiology. So I'm familiar with pathogens, but not environmental microbes. Yeah, I see. Maybe I see the 8. Let me zoom in first. It is one of the survivor. One ciliate. You can see the size is larger than bacteria. Although bacteria is longer than the ciliate, but the overall the volume ciliate is larger. And it is very easy to tell whether they are bacteria or other ciliates. Yes, I have a job, sorry. I only do this after my work. And in the middle, you can see a dead body of a ciliate. It is not moving, but you can see it's very clear that it is a ciliate dead body because it looks like a cell. How did a leaf look like under the microscope? Let me see. I just have some I need to find, but because I'm not quite sure whether I have a leaf sample. I have a moss sample. Maybe we can look at this. I have a moss sample. I add water and mix with the moss. And because I want to find water bear, so I prepared this. I think 2 days ago. Okay, maybe I will put the moss sample under the microscope. Let me put down my phone first. By the way, maybe I will start a new live session on Sunday because I want to prepare the day 0 of the new sandbox together. Maybe you can tell me what you want me to add into the new sandbox first in the comment section. Or under the comment section of my day 100 video. Dead skin and human hair. Okay, maybe I will look at this later. It is a 3 days old moss samples I've prepared. And you can see there are some. I need to zoom in first because I don't know what it is. It has a very weird motion. I'm not quite sure what it is. Maybe paramecium, but I think a little bit different. Yes, actually, active microbes swim very fast. Sometimes if you don't want them swim very fast, you can add some like albumin. It will make the sample more viscous and they will swim more slowly. Maybe I start from the top to look at them. And sometime, or you can see in many of my videos, some rock structure like this. It is actually some types of fun algae. And this is very common, like in the fresh water. Maybe we can look if we can find water bear in here. Actually, I found many dead body of water bear, but I can't find any alive water bear. Can you create a super serum? What is super serum? Yes, I did. I think 2 days ago I still found some water bear, but not alive. They are not alive. They are just dead body. They are not moving. I'm sorry, I need to prepare something first. I think I see something here. It is Perenema. I can type it in the comment section. I think I found this microbe in some days of the sandbox. And you can clearly see that there is a flagella moving very active in front of it. Yes. And actually this flagella not only help them to swim, and actually it can use the flagella to detect whether there are prey in front of them. What if I put a fly? I don't know. I never put a fly inside the sandbox. Maybe we can do it in a new sandbox. And you can see there are many hair-like structure on the cell. Actually, it is cilia. they use the cilia to help them to move because they have cilia. So that's why they are called ciliates. Actually, any microbes that have cilia, they will be called ciliates. So that's why I mentioned a lot of time in my videos like ciliate, because a lot of microbes are classified as ciliate. I think it is Euplotes. EUPLOTES. It is also one of the most common residents inside our samples previously. No, I didn't put any sperm on the slide, so no, it is not sperm. Actually, sperm is also one of the most common. Oh, yes, sperm is also one of the most common requests. And can you see in the middle there's a large debris here? It is a head of a mosquito larva. Just a head. I don't know why. It is capitated. You can see the black spot is his eyes. Yes, I think there are structural similarity between human cilia between human and ciliate, but I think the molecular structure, molecular details are quite different. I think I just saw some nematode, but I didn't stop to look at them. Maybe we can find some to look later. Because I took the moss together with some soil, nematode is very common inside the soil sample, so you can see many nematode inside. And I think one of you asked me to look at a leaf, and you can see it is a moss leaf. You can see each individual plant cell very clear. And in the middle, can you see a circle shadow? It is actually a testate amoeba shell. Some amoeba has shells, but some doesn't. Okay, maybe we keep going. And can you see in the middle, it is definitely a ciliate, a very large ciliate, but it doesn't move anymore. But you can see a very large vacuole inside the cell. I think it is that. Because you never see a ciliate not moving like this. Even they are not moving, you can still see the cilia moving. I think there are more microbes in the more sample than my sandbox. And this is a dead leaf, so you can see some cells still have chloroplast, but most of them is empty. You can see in the plant cell there are many ball-shaped structure. It is chloroplast. And most of the plant cells are empty because they are dead. And for the newcomer, I plan to have a live session on Sunday, and we will prepare the new samples together. So maybe before that, I need to prepare some materials. If you want me to add any materials, you can comment in my day 100 video so that I can prepare earlier. Otherwise, if you mention some rare materials, I can't prepare it. Okay, in the middle, you can see, I think it appeared in our samples before. It moves so fast. Yes, here. It is a ciliate and it looks like a kidney. Can you see its shape? It is very like a kidney. It is actually called copoda. If it moves very fast, I cannot focus. Maybe we will use a lower magnification. Yes, this. Yeah, copoda is a very common ciliate that's living in dirty water because they like eat E. coli. So if you leave some water in the toilet for a long time and you put them under the microscope, you can see many Eupoda. Okay, it stopped here, so I can focus it. And you can see they use the ciliates to create water flow, and many materials flow inside the body so that they can capture and eat them very convenient. And for unwanted things, you can see that they expel it very quickly. And they eat E. coli. They like to eat E. coli. Yes, also it looks like a very early stage embryo. And you even can see their cellular structure are moving inside, and most of them are actually food storage. So actually, the number of copoda can tell whether the water is dirty or not. The more the number of copoda, it means the water is very dirty. And here it is a very active nematode. And you can see compared to other ciliates, its size is very large, much larger. And for information, this bubble, yes, this circle-like structure is actually air bubble. Yes, it is a nematode. And actually, there are a lot of nematodes living inside soils. So whenever you touch soils, it means that you are touching a lot of nematodes. But the good news is most of the nematode is not harmful to human. They cannot cause any disease in your body. But some species can actually cause disease, so you still have to be careful. Actually, nematodes still have some simple structure of like intestine. So they will ingest anything like microbes smaller than them, and they will digest it in the intestine. Yes, it is also one of the reasons why you have to wash the vegetable before you cook them. And actually, some nematode do not just infect you by do not just infect you by eating them. Some nematode can penetrate your skin. So the best way to prevent infection is that to prevent directly touching soil, because some of the nematode can secrete enzyme to digest your skin cells and penetrate your skin. And I'm now using 100 magnification. Oh, I've seen a water bear here, but I think it is not moving. So I can't tell it is alive or not. Actually, I saw many I saw many water bear in the moss sample, but I seldom saw any actively moving water bear. What do you mean? You mean those microbes swimming around the water bear? But unfortunately, I cannot tell the definite identity of these microbes because I can just tell you that they are serious, but I can't tell you what it exactly is. But I think there's a very high chance that this water bear is dead already because you can see there are many bacteria surrounding it. Maybe it is going to degrade yes, it's going to degrade the water bear. You can see near the mouth of the water bear, there are many bacteria surrounding there. Yeah, you can clearly see that there are much more bacteria surrounding the mouth of the water bear. Maybe because the skin of the water bear is tough, it is difficult to digest, but bacteria can digest the water bear from the fragile part like the mouth. So anyway, I think this water bear is dead already. I think I need to prepare some fresh moss sample. Because I have put aside this moss sample for 3 days. It incubates a lot of bacteria to survive inside and proliferate. So that's why the water quality inside the moss samples become worse and worse. What is my most active samples? I think I actually had a moss sample that I have prepared for 10 days before. It really contains a lot of microbes inside, but I threw it away already. By the way, I want to know what time is it in your place? Like, do you have to sleep or go to work? Or go to school? Okay, in the middle, you can see I think it is also a dead body of a rotifer. It is a rotifer, but I think it is dead already. Because I'm trying to figure out what is the best timing to hold the live session. Okay, I think I have go through the whole slide already, so I think nothing special in the moss sample. Maybe we can look at the old sample of the samples. And let me take them out first. Sorry, I put it back here. Why the every water bear are dying? Because it is a old moss sample. So it means that the water quality inside the mold sample is getting worse and worse already because the bacteria are proliferating inside. So if I want to find some live water bear, I need to prepare some fresh mold sample. And I find out some tubes, backup tube from a long day before, day 42, day 43, 45. Because I prepare backup tube every day, but some of them I still haven't filled up. Which day do you want to look at? By the way, you may be curious about why day 42 is so milky. It looks milky because we added baby oil, I remember, and it kills everything. You want to look at day 42, okay. And you can see at the bottom, it becomes red. Actually, it is also a kind of algae. And you can see the wall of my sandbox also turns into red. It is also due to the action of some algae. What the latest one I have collected, day 99. The latest one is day 99. I mix it well first. I can't disturb the sediment under the bottom. It sticks so hard under there. Anyway, we take some out first. Do you plan to sell this sample? I don't. It is just dirty water. I think it doesn't worth any money. And also, may I know, is it really legal to sell this type of water? Would it be counted as bowel weapon? Like you are selling pathogens all around the world. I think there are nothing surviving inside the day 42 tube. There are many debris here, but I don't see anything moving. Maybe zooming first. Yes, you can see even bacteria, they're also not moving. So I think everything is dead inside here. So maybe we look at the other tubes. Okay, so I think the day 42 tube can be discarded. And day 43, it looks better because I'm not sure whether there are other lives like bacteria or microbes, but at least there are algae inside because it is easy to tell because it looks green. And we can put it under microscope. Sorry, what do you mean? What do you mean? You want to eat what? You mean the old tube? You mean the content of the old tube? Let me check well first. Okay. And actually, it is not a good practice because never try to shake anything that is suspected to contain a lot of bacteria because it is just helping them to spread away. So if you are really doing this stuff in your place, it better have protection like wearing masks. Actually, you still cannot see anything moving, but it is normal because it is a very old tube. And you can see many bubbles. We can look at this. You can see many bubbles. Some are green. And for those green purple, you can see you can see some content inside. Actually, you can tell it is actually algae, but there are also some bubble like in the middle. It is very clear. You cannot see anything inside the bubble. Actually, it is oil droplets. Because it is day 45 and day 42 we have added baby oil. So maybe it is just leftover baby oil inside. So it is a oil droplet. But the green one are algae. And in the middle, the empty circle is all droplets. And now you can see there are more green color circular stuff. It is algae. So when you look into a sample, when you see something like a bubble, the strategy to tell whether it is a bubble or an organism is to look inside the bubble, whether there are content inside. If there are content inside, it is a cell, not a bubble. Yes, this tube just contains algae, but nothing else. And actually, there are many other types of microbes, and I think maybe we have chance to grow them in the new sandbox. And for examples, algae, most of you may think algae they are not able to move, but actually many algae can move. They can swim. I think it would be also interesting to grow some motile algae inside the sandbox. What is the latest collected sample? You mean the day 99 samples? Yes, the day 99 actually, there are some paramecia, but not many. I can find it out first. Let's just give me a minute. I have day 99 sample. If you want, we can take a look. I'm not quite sure because day 99, if you go back to my video, you can see most of the microbes inside the samples were not very active. One of the reasons why they are not very active because I haven't diluted the samples for a very long time, like over a week. What if it is active now, and then it will be good. Let's check it first. Maybe there are some, I don't know. Um, yes, actually it just looks like day 100 samples. They are just bacteria, but I can't see any other microbes. Yes, there are dry ash there. We added on day 100, I remember. Yeah, let me zoom in. And this brown object is the shell of testate amoeba. So as you can't see it is moving, so it is very likely that it is dead already. So I mentioned that testate amoeba can build their own shell. So as the shell is made of calcium, it is very hard to be degraded. So even the amoeba is dead, the shell will be left in the samples for a very long time. I still can't see anything. And speaking to living material, I just remember I have some tree sap. Tree sap. I still have some tree sap. Maybe we can look at this. I even can't see any dead body of Paramecia. Maybe they are even be degraded by bacteria. Yeah, I see something here. Maybe we zoom in first. Okay, it is. It also looks like a testing amoeba shell, but it is a very young testing amoeba. You can see the shell is transparent. You can see the content inside. And the shape is quite different than the previous one we saw before. It would be also a good idea if we can arrange a party and we drink together. It's unfair for me to drink alone. And this shell is partially degraded, so you can see it is transparent. But anyway, I still can't see any interesting microbes, so maybe we stop here. If you want to see some interesting sample, maybe we can look at this, a tree sap sample. Here, a tree sap sample. But I can't promise whether there are really interesting microbes inside. So we just have a look first. Hello. Are you Japanese? I saw your name is Japanese. You mean the sound? Sorry, I just mixed up. It is not the one. Um, I don't know, but I see in the middle it is a nematode, a pretty small one. And you can see, yes, of course there are some things swimming. It is a very, very small ciliate. It is around the size of bacteria. But apart from that, you can see many oval-shaped material. Can you see it? Some are brown, some are slightly green. They are like a rice grain. Actually, it is mold spore. Some mold produce the spore here. For the brown one, actually, they are probably the spore of black mold. So the spore are brown in color. So maybe you will wonder how we can tell it is a mold spore or it is a ciliate or other microorganism. You can see that like in the middle there's a brown spore. It is oval in shape and you can see there are septate inside. So it is very common for mold spore to have septate inside. So when you see some oval in shape and you can see there are some septate, it is very possible that it is a fungal spore. Yes, I can still see something moving, but they are very small, not very large one. By the way, I'm using 400 magnification now. Because I'm looking at old samples so far, so yeah, maybe they originally very active, have a lot of life inside, but now they may be die already because I've left aside for a long time ago. Yes, it is a tree sap, not a sandbox. Yes, I need to get some new samples. So maybe I will prepare it on Sunday and after I prepare them, I will hold a live session. We can look at them together and put it into the new samples as the starting material. I think, okay, in the middle, you can see clearly an organism. It is a mite, but and also you can see there are eggs inside the body, so it is a pregnant mite. And when we are talking about mites, you may concern about whether it is a pathogen to humans, but actually in soils or like natural material like tree saps, it is very common for some mites living there. So they're actually not necessary to be harmful to human. It is not a spider, it is a mite. You can tell it from its structure. Actually, mites and spider are very close in genetics. And you can see there are eggs inside. Yes, you can see the eggs inside the body, but I think like the mite has already died and there will be no chance for the egg to hatch already. And how to differentiate spider and mite? You can see the mite, it has a very clear head here, a very long head here. This one. And for spider, their heads are not like this. And also for spider, there are also more than 2 eyes. And for mite, they have just 2 eyes. Okay. And remember we will hold a live session on Sunday. I need to prepare some material before the live session. If you want me to add anything inside the samples, you have to tell me earlier, like leave a comment under the day 100 comment session so that I can prepare it earlier. Or otherwise, I cannot, for some rare material, I cannot suddenly get into it then. Okay, it is a ant. But it is obvious that it is dead already. Yes, Sunday. I will hold a live session on Sunday, maybe the same time like today or earlier. So wait a minute. You want me to add bacon? Okay. Continue. For information, if you want to look at an ant like this, it is a 4T magnification. For 4T magnification, it would be so small that you cannot observe any microbes. Unless the microbes are very large, like Paramecia. I don't think there are anything interesting here anymore. Or if you want, you can look at the balls I prepared for growing water bear before, because there are still many microbes inside. Okay, anyway, I don't think I see any interesting microbes inside the tree sap, so maybe I stop here. And before I end the live session, maybe we take a look at the water bear boss first. Although I have added around 20 water bear inside, but I'm not quite sure whether the water bears grow very well inside the boss. So we just look at this, but I can't promise whether they are really water bear. So it is maybe it is one of the beauty of microbiology, the uncertainty. And that's why it is interesting to do the sandbox projects. The uncertainty is very interesting. You can't even predict what will appear before you look into the microscope after adding anything inside the sandbox. And they change very frequent. Once you change the environment, the microorganisms inside the sandbox will also change. Okay. It is the last thing I would do before I end the live session. Let's look at the boss I prepared for water bear. But I found that there are many microbes inside this boss other than water bear. Yeah, there are many microbes inside this boss, but I'm quite sad that I still can't find any water bear living inside. Even though I have added 20 water bears inside, but I can't find them out anymore after that. But actually the boss is large. It is impossible for you to find these 20 water bears out for a short time unless they reproduce. What I want to do is that they can reproduce inside the water bear boss, but I don't think they are now reproducing very well. I want them to build a water bear kingdom. Yes, they can reproduce. Water bear can lay eggs, and they don't have to mate. They can lay eggs by themselves, even there are only one water bear. In the middle, you can see a Halteria. It is also called a jumping ciliate. You can see instead of swimming, it looks like it is jumping. And in the middle, you can see a microbes. Although it is smaller than a paramecium, but actually it is a polycellular microorganism. So actually you cannot judge whether it is a polycellular microorganism or single cellular microorganism just based on the size. A single microorganism can also be very large. Yes, theoretically, they are very good against natural disturbance, but in reality, they are very sensitive to the environment, actually. Especially when the environment, when there is sudden change in the environment. If you heat a water, if there is a cup of water and there are water bear inside and you heat them to 100 degrees Celsius within a short period of time, like few minutes, they will also die because they don't have time to enter dormant stage. You have to give them time to enter dormant stage before they can be go against the high temperature. If you heat the water for 1 hour from room temperature to 100 degrees Celsius, then maybe they will have time to go into dormant stage and they will survive. So the change cannot be so sudden, actually. And it is a good question. Actually, it is possible. Whenever you change the environment, actually you are giving them a selection pressure. Some microorganisms that are adaptive to the environment, they will survive, and for those who not, they will die. So when you constantly change the environment, you can actually select what microbes are suitable to survive inside the environment. And also microorganisms, their genetic is very unstable. Unlike human, our genetic is very stable. So actually microorganisms, they can evolve very rapidly. And you can see in the middle there is a very weird motion polycellular microorganism. Actually, it is a waterfall also, but it is a different species. A different species. Actually, there are a lot of species of waterfall. It is a tough question because I really don't want to be a microorganism. If I want to have the ability, maybe actually microorganisms do not have many abilities. So maybe I took water bear. At least they have the ability to survive under a very bad environment. So maybe in the future, if there are any apocalypse, I can survive. So you can see that microorganisms tends to live near some debris because some microorganisms they like standing on some objects. They don't like swim. They like standing on objects. Like this, it is Euprotes. I think I have mentioned it before. It looks like a cockroach, a microscopic cockroach. It uses its cilia. It uses its cilia to walk instead of swim sometimes. So in the middle, you can see what what I described is a microscopic cockroach because it, instead of swimming, of course, it still can swim like this. It is a swimming Euprotes. But if it want to walk, it is also able to walk on an object, like a cockroach. What is that stuff or debris in the water? I don't know. Maybe dead plant cells or just mud. I'm not sure. Because apart from microorganisms, I didn't add any animals inside the box, so it would not be any feces of other animals. What would be the optimum temperature when I saw the most? I'm not quite sure because most of the time I just put the sandbox or the water bed box under the ambient temperature. I didn't put them into the incubator because I don't have an incubator in my home. Of course, it would be great if I have an incubator because most microorganisms, for most microorganisms, their optimal temperature would be like 37 degrees Celsius. You mean what? You mean no? You mean what? You mean the circle things? You mean this? The coins like things? You mean this? It is also a testate amoeba shell, but this one is alive. It is not a dead testate amoeba because I think this testate amoeba is still very young. The shell is transparent. You can see the body of the amoeba inside the shell. Can you see inside this circle? There is also a circle inside. And you can actually see 3 layers of circle. The outer one, the middle one, and the inner one. The middle one circle is the cell membrane of the cilia of the amoeba. And you can see it moving inside actually, but not very fast. You can see it move very slowly. Yes, the light yellow one, yes. And if you look into it very carefully, you can see its intracellular structure are changing, and you can see it move very slowly. Amoeba never walk very fast. They walk very slow. You really need to pay attention on this motion. Actually, I can, but I need to change the objective because I take it away. I have a 40 magnification objective. It can zoom in into 600. I need to change the objective first. Sorry. I can zoom it further actually. No, no testate amoeba is deadly. All the testate amoeba is harmless to human. So whenever you see a amoeba that has shell on it, it is not harmful to human. Most amoeba that are harmful to humans are naked amoeba. They don't have shells. Okay, you can see it slowly going away. I just plug in a more powerful objective. We can look into a 600 magnification. But also the image quality will be lower. The higher the more it magnifies, the poorer the quality of the image. Okay, maybe we look into other microorganisms. Okay, for those who are new here, I want to announce that I would hold a live session on Sunday because I want to prepare the new sandbox together. So if you want me to add anything on Sunday, please leave a comment on the day 100 video to tell me what you want me to add so that I can prepare earlier. Okay. It is a dead leaf. You can see a dead leaf is also a paradise for microbes to survive and live. So you can see there are many microbes inside. Let me see if I can zoom in. But most of them are living on the bed of the leaf, so I cannot see them clearly. Again, it is a Halftonia. You can see it is a jumping cilia. So instead of swimming, you can see it jumping. You mean what? Yes, I think I see some paramecium living on the leaf. But they are behind the leaf, so I cannot see them clearly. But anyway, it's okay. Yes, they looks like a jellyfish actually. So this one, it is a older testate amoeba. You can see the shell is actually much rough. It's rough, and it is not that transparent. You cannot see its inner structure clearly, but I can tell it is dead already because you can see something moving inside. It is actually a ciliate going inside because once the testate amoeba die, they leave the shell here and they left the shell here undegraded. It is a best place for some ciliates to live inside. So some ciliates will use it as their home. Okay, in the middle you can see a naked amoeba. This one walks very fast. You can clearly see it moving very fast. It was like a liquid. Is this amoeba deadly? It is impossible to tell whether this amoeba is deadly by just looking at the microscope. But because some amoebas are able to go into your brain, like an Naguaria fowleri, also known as brain-eating amoeba, but based on this morphology, maybe it is not a brain-eating amoeba. But amoeba is actually very common in fresh water. Most of the species are not harmful to humans, so I guess it is not. For brain-eating amoeba, they have special structure, so it is. If I see them, maybe I can identify them. But actually, you eat the brain-eating amoeba, you are not likely to be infected because it is very, very rare for brain-eating amoeba to infect humans. For every year, there are only few cases in the world to have any people got infected reported. Oh, you are the guy asking for the tree bark. Okay, maybe I will add this as the starting material in the new sandbox. Can you leave the comment in the day 100 video so that I can remember? Or maybe I drop it down too. Okay, maybe we will add tree bark in the new samples. Thank you. And for the newcomer, we are looking for water bear if there are really water bear inside. Okay, in the middle, you can see testate amoeba, but with a different shell shape. It is also alive, you can see. You can see the inner structure. It is moving. Yes, it is. It looks like a sun. Even the same species of amoeba, they will make a different shape of the shell. It is testate amoeba. Testate amoeba means any amoeba that has shells. They are able to make a shell for them to as a home and protect themselves. Oh, it is not. It is not something below it. It is actually something inside it. Because the shell is not very mature, it is transparent. You can see clearly the amoeba inside the shell. So you can see many bubbles inside, and you can see the bubble inside is moving. It is actually the inner structure of the amoeba. And you can see these two ciliates here. It is surrounding a debris. Maybe it is digesting the debris. What is the shell made of? It can collect any materials inside the environment, but in majority, they will collect something that contains calcium to form the shell. And of course, it can build the shell based on any components inside around the environment. It really depends on what is inside the environment. So the shell component can be different. I worked as a microbiologist in hospital. My job is to identify any pathogens inside clinical sample and give report to patient. And also suggest any antibiotic or drugs that can be used to kill the pathogens. I think it is a paramecium, but it is hugging a debris, so I can't 100% sure whether it is a paramecium, but it is very likely. Oh, it would be good if you do it for your science fair, but be careful, don't get infected. And compared to the previous testate amoeba we saw, you can see that this shell is empty. It is very easy to tell. You can see there's nothing inside. You cannot see anything moving inside. Yes, all you need is a microscope. Actually, you can buy, if you don't have a lot of money, you can buy some cheaper amoeba from Amazon or eBay. Even some few hundreds US$ microscopes are enough to observe microorganisms. My first microscope only cost about maybe 100 and maybe 200 USD. And whether the microscope or expensive or not really depends on what you want. Actually, if you want a very high image quality or some advanced image technology, you may need to buy a very expensive microscope. Or if you're just a beginner, maybe you don't really need so advanced technology, you can just buy a cheap one. My microscope costs about 3,500 USD. Against a naked amoeba. This sample is from my water bear jar. I built a water bear box. I built the box just for growing water bear, but accidentally it grows other microbes other than water bear. But it is not my goal actually, but I don't mind. Actually, it is also good. Okay, I think I can't see any water bear here, so maybe we can stop here. No, I can't find any water bear. But we did find a dead body of a water bear in a moth sample today. But I think it was a dead body because it was not moving anymore. All ad requests should be commented on the day 100 video we will be testing. Yes, I think I will write a post later. I think I'm going to end the live session soon, and I want to repeat again, if you want to, because we are going to hold a live session on Sunday, if you want me to add anything inside the new sandbox, please leave a comment on the day 100 video and tell me what you want me to add as a starting material. And I will prepare it before the live session, if possible. If I cannot get the material before Sunday, then maybe I will add them later. And also, I have a question. Because my first proposal is because we are going to work with the sandbox in 2 jars, and I have mentioned one will be put under the dark, one will be put under the light, always under the light. And I want to ask, what do you think? Like, should I put identical stuffs in the 2 samples, or I put different things into 2 jars? I'm not quite sure actually. And also, do you think it is necessary to put them into separate environment, like under the dark or light, or maybe they can put under the same environment, like under the light, both jar under the light? It is okay. I'm trying. Maybe I will set a poll, and maybe you can vote what proposal you prefer. Because I haven't decided yet. But the problem is once I started, it is difficult for me to change anymore. So I better to decide it before day 0. You mean on day 0? Yes, I think I will put identical stops inside the 2 jars in day 0. But starting from day 1, I will put different things into the 2 jars because I want to see how they progress differently when I add different things inside day by day. So I don't know. Maybe I set a pool to decide, and maybe you help me to vote. I have some choices, and you help me to vote what proposals you prefer, and I will start with them. Okay, maybe I will end the live session here. I will start the live session on Sunday around the same time like today. So yeah, if you have time, you can join the live session on Sunday. Oh, it doesn't matter because we are going to hold the live session on Sunday again. We will prepare the new sandbox together. So again, if you want me to add anything inside the new sandbox, please leave the comment under the 100 days, day 100 sandbox video and tell me what you want me to add. Thank you. Goodbye.